Hello everybody, how are you? Hope you are all well. I am Dr. Dua Karim Metwalli, lecturer of histology. In this video, we are going to continue the second part of the histology of the eye. The third layer or coat of the eyeball is the inner nervous layer or coat the retina. It is a modified nervous tissue lining the innermost layer of the eye. It is posterior. It is the posterior 3/4 of the eyeball inner to the choroid. Histologically, the retina is formed of 10 layers, and they include the retinal cells or cells of the retina, which are retinal pigmented epithelium, retinal nerve cells like the photoreceptors, bipolar cells, ganglion cells, and association neurons like horizontal and amacrine cells, neuroglia, molar cells. All these cells found in the 10 layers of the retina. Another classification of these cells may be the photoreceptor cells, the retinal rods and cones, conducting neurons, the bipolar neurons, and ganglion cells, association neurons, horizontal cells, and amacrine, centrifugal, interplexiform cells, supporting cells like neuroglial cells, molar cells, and microglial cells and astrocytes. What are the layers of the retina? Let's now know the layers of the retina. The first layer is a layer of retinal pigmented epithelium. Then a layer of rods and cones, the photoreceptors. The external limiting membrane layer. The external limiting membrane layer. The outer nuclear layer, then the outer plexiform layer, the inner nuclear layer, then the inner plexiform layer, then ganglion cell layer, optic nerve fiber layer, and finally the internal limiting membrane layer. Developmentally, the retina is formed as a downgrowth of the forehead ectoderm, which invaginates to form the optic vesicle. Then invagination of the optic vesicle makes the double-walled optic cup. The outer wall is thin and forms the pigmented epithelium, retinal pigmented epithelium, while the inner wall is thicker and forms the neural retina. So this is the outer wall, formed only from the retinal pigmented epithelium, while the inner wall formed of the rest of the layers of the retina, as we see. Okay, again, the cells of the retina include retinal pigmented epithelium, retinal nerve cells, which include the photoreceptor cells, roots and cones, bipolar cells, and ganglion cells, association neurons like the horizontal and amacrine cells, neuroglial cells, molar cells. These cells are arranged in the 10 layers of the retina, which are the first layer, retinal pigmented epithelium, then layer of photoreceptor rods and cones, external limiting membrane, outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglion cell layer, optic nerve fiber layer, and finally the internal or inner limiting membrane. Another classification of the cells of the retina is the photoreceptor cells, include the retinal rods and cones, conducting neurons responsible for a conduction nerve impulse, bipolar neurons and ganglion cells, association neurons, horizontal, centrifugal, plexiform, and amacrine cells, horizontal and amacrine cells. Supporting cells, neuroglial cells like molar cells, microglia, and astrocytes. Another light microscopic photo showing the layers of the retina. As we see here, this is the layer of retinal pigmented epithelium, then layer of roots and cones, the photoreceptor cells, then the outer limiting membrane and the outer nuclear layer, the outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglion cell layer, layer of the optic nerve fibers, and finally the layer of the inner limiting membrane. This diagram shows the different types of the cells. This is a retinal pigmented epithelium, and these are the photoreceptor cells, rods and cones, and these are the bipolar cells, and this horizontal and amacrine, horizontal cells and amacrine cells, and these are the ganglion cells. And these tall cells, these cells, two sides, these cells are the molar cells. This cell, tall and columnar cell, these are the molar cells. Okay, let's start with the first layer of the retina, the retinal pigmented epithelium. It is the outermost layer of the retina. By the light microscope, the retinal pigmented epithelium appear as a single layer of cuboidal cells resting on a basal lamina. This basal lamina is a part of the proximal membrane of the choroid. The cells contain basal rounded nuclei, and they have basophilic cytoplasm containing melanin granules. By electron microscope, the retinal pigmented epithelial cells shows the apical surface shows microvilli, and these microvilli contain melanin, as we see in the picture, and these microvilli surround the outer segments of the photoreceptors, the rods and cones. The basal plasma membrane show enfoldings with numerous mitochondria, and this is a character for ion transporting cells. The lateral borders show junctional complexes and gap junctions, and the cytoplasm by electron microscope also shows mainly smooth endoplasmic reticulum, lysosomes, and residual bodies. It also contains melanin and lipofuscin pigments. Let's know how the retinal pigmented epithelium adapted to its function. The function of retinal pigmented epithelium. The cells contains melanin or pigment granules, which responsible for absorption of light to prevent light reflection after the photoreceptors stimulation. Also, they are responsible for nutrition of the photoreceptors and esterification of vitamin A by the smooth endoplasmic reticulum they contain, and they transport the vitamin A after esterification to the rods and cones. Also, they are responsible for phagocytosis of old discs extruded from the tips of rods and digest them by their lysosomes. As we mentioned that the cells have lateral junctional complexes between the cells, and these form the outer blood retinal barrier to protect the retina from harmful molecules in blood vessels of the choroid. This diagram shows the functions of the retinal pigmented epithelium and how it is adapted to its function. The light enters the eye and first received by the first layer of the retinal pigmented epithelium, and then goes to the other layers. The melanin granules absorb the excess light to prevent light reflection. As retinal pigmented epithelium contains the melanin granules, resulting in absorption of light and prevent reflection after photoreceptor stimulation. Also, retinal pigmented epithelium is responsible for nutrition of photoreceptors, as it is close to the underlying choroid and blood vessels in the choroid. Also responsible for esterification of vitamin A by smooth endoplasmic reticulum, and transport to rods and cones. Phagocytosis of the debris or old discs extruded at the tips of the rods and digest them by their lysosomes. Formation of the outer blood retinal barrier by the lateral junction complexes between cells to protect the retina from the harmful molecules in blood vessels in the choroid. As we see in this diagram, shows how the light enters and received by the retina. The light enters and passes through the eye and received by the first layer of the retinal pigmented epithelium, and then pass through the photoreceptors and the other layers of the retina, and then pass through fibers of the optic nerve and enter through the optic disc. And this is the blind spot. Contains no photoreceptors, so it is a blind spot. Okay, the second layer of the retina is the layer of rods and cones. This layer contains only the outer segment of the rods and cones. Outer segment of the rods and cones. Now let's know the structure of the rods and cones. The rods and cones are polarized neurons. Each one has a single photosensitive dendrite at one pole, while the other pole synapses with bipolar cells. They are formed of outer segment, inner segment, cell body with nuclear region and synaptic region. Outer segment, inner segment, cell body with nuclear region and synaptic region. Nuclear region and synaptic region. Okay, let's start with rod cells. The rod cells are about 120 millions. They are sensitive to the dim light. They consist of outer segment and inner segment, and they are connected at what is called connecting stalk, and also consist of the cell body with the nuclear region and synaptic region. The outer rod segment is cylindrical in the shape, embedded in between the microvilli of retinal pigmented epithelium. The outer segment of the rod contain membrane bounded discs, membranous discs arranged in stacks, and they are not connected to the cell membrane, separated from the cell membrane, not connected to the cell membrane. These discs contain the visual pigment rhodopsin. The connecting stalk, which connects the outer and inner segments, it is modified cilium formed of 9 doublets without central singlets, 9 doublets of microtubules, doublets without central singlets. The inner segment of the rod is a protein sensing part. The outer part ellipsoid and contains mitochondria, and the inner part called myoid contains the rest of the organelles, Golgi apparatus, rough endoplasmic reticulum, and ribosomes. This segment is responsible for sensing of the visual proteins, and then pass to the discs in the outer segment. So new discs are added, and old ones are phagocytosed by the retinal pigmented epithelium. The cell body of the rods include the nuclear region and synaptic region. The nuclear region show the nuclei of the rod are small, rounded and dark. They form the nuclei of the roots photoreceptors form the majority of nuclei in the outer nuclear layer. Synaptic region in the form of spherule lie in the outer plexiform layer. Make synapse with the dendrites of the bipolar cells and axons of horizontal cells. The cone cells are about 6 million, and they are responsible for the color vision and the high visual acuity. As we see here, they have the same structure as the rods, but with some differences. The cone cells differs from the rod cells in the outer segment is conical in the shape. The membranous discs are continuous with the cell membrane. The discs contain visual pigment edopsin. The nucleus is larger and less dense. The synaptic region is in cone pedicle. The cones are formed of 3 types. Each is sensitive to certain color, red, blue, or green. The third and the fourth layers of the retina, the external limiting membrane, and the outer nuclear layer, external limiting membrane, and outer nuclear layer. The external limiting membrane actually is not true membrane. It is formed of row of tight junctions between molar cells with each other and rods and cones. These are the molar cells, and these are the outer parts of the molar cells, and this is the inner parts. The outer part of the molar cells is responsible for formation of the external limiting membrane, while the inner parts, the bases, form the internal limiting membrane. So the external limiting membrane, as we mentioned, is not true membrane. It is formed of row of tight junctions between the molar cells with each other and with the rods and cones in between. The outer nuclear layer, as we mentioned, formed by the nuclei of rods and cones, and also, as we mentioned, the majority of the nuclei in the outer nuclear layer formed of nuclei of rods mainly. The outer plexiform layer formed of synapse of the rod spherules and cones, pedicles of the cones, with the dendrites of the bipolar cells and processes of the horizontal cells. The inner nuclear layer, this is the inner nuclear layer. As we see, it is formed of the nuclei of the bipolar cells. This is the bipolar cells, and these are the nuclei, and nuclei of the horizontal cells and amacrine. This is amacrine cell, and nuclei of molar cells. Molar cells. So the inner nuclear layer, again, formed of nuclei of bipolar cells and nuclei of horizontal, macrine, and molar cells. The cells of the inner nuclear layer, as we mentioned, include bipolar cells, horizontal cells, amacrine cells, and molar cells. The bipolar cells or neurons, their dendrites synapse in the outer plexiform layer with the rods and cones, and their axons synapse in the inner plexiform layer with ganglion cells. The horizontal cells, the association neurons, synapse with the rods and cones. Synapse with the rods and cones in the outer plexiform layer. Amacrine cells, they are also association neurons, like axons, synapse with bipolar axons and bipolar axons and dendrites of ganglion cells in the inner plexiform layer. The last cell is the molar cells. Molar cells are neuroglial or supporting cells, forms the inner and outer limiting membranes. Their outer ends join the rods and cones by junctional complexes, forming the outer limiting membrane. Their inner ends form the inner limiting membrane. The inner plexiform layer formed by the synapse between the axons of the bipolar cells, dendrites of the ganglion cells, and the processes of the amacrine cells. The ganglion cell layer formed of the cell bodies of the ganglion cells, which are large multipolar cells. The optic nerve fibers layer formed of the axons of the ganglion cell. These axons of the ganglion cells. They are unmyelinated nerve fibers covered only with myelin after they exit from the sclera. So the optic nerve fibers layer formed of axons of the ganglion cells. The internal limiting membrane, as we mentioned with molar cells, formed of inner ends or processes of molar cell and their basal lamina, and it separates retina from the vitreous. There are specialized areas in the retina, include the macula lutea with the fovea centralis, and optic disc, which is also called the blind spot or optic papillae. The macula lutea lies opposite the center of the lens. It is about 3 millimeter in diameter, contains yellow pigment, hence its name, lutea equal yellow. It has high concentration of the photoreceptors and increased number of ganglion cells. The fovea centralis is a central rounded depression in the macula lutea, This part characterized by that the layers from the outer nuclear layer inward are displaced laterally, displaced to the lateral side, displaced laterally, as we see in the picture, from outer nuclear layer inward, displaced laterally. This area contains only the cone photoreceptors, and the cones present are rod-like. And this part also leak blood vessels and nourished by the choroidal circulation. The fovea centralis responsible for the accurate visual acuity. The blind spot or the optic disc is a site where the ganglion cell axons converge and exit from the eye. It has no photoreceptors, so it is insensitive to the light. The retinal blood vessels enter and exit through it. The blood-retinal barrier. The retina has two blood barriers. The outer barrier, outermost, is formed by the tight junctions between the retinal pigmented epithelium, and it limits escape of substances from the choriocapillaries to the outer part of the retina. The inner barrier, the inner part of the retina, protected by tight junctions between endothelial cells of the retinal capillaries from the central retinal artery. So the retina has two blood barriers, the outer and the inner. The outer formed by tight junctions between retinal pigmented epithelium, while the inner formed by the tight junctions between endothelial cells of retinal blood capillaries. Okay, now let's discuss the eyelid. The eyelid is movable folds of the tissue that protect the eye. Protect from injuries and excessive light, also help in spreading the tear film over the cornea and conjunctiva, and also help in drainage of tears by the lacrimal system. Let's now know the structure of the eyelid. This movable fold of tissue formed of anteriorly, then posteriorly. Anteriorly, skin and subcutaneous areolar tissue. Then there is a layer of skeletal muscle, striated muscle, orbicularis oculi. Then dense connective tissue layer, the tarsal plate, and finally posteriorly the palpebral conjunctiva. The structure of the eyelid, as we mentioned, formed of skin, orbicularis oculi muscle, and tarsal plate and palpebral conjunctiva. The skin is loose, elastic, with no subcutaneous fat. 3 to 4 rows of eyelashes project from the lid margin, but without erector pili muscle. It also contains modified sweat and spacious glands. The orbicularis oculi muscle, skeletal muscle that surrounds the eye. Then the tarsal plate. This is a tarsal plate. It is a dense connective tissue containing meibomian glands. These are the meibomian glands. They are modified species glands and open at the three edge of the eyelid. These glands secrete oily substance that becomes incorporated into the tear film, preventing evaporation of the tears. The last part is the palpebral conjunctiva. This is the skin and muscle, tarsal plate, and the conjunctiva. The eyelashes are about 100 in upper lid and 50 in lower lid. The upper lid lashes are directed upward and outward, and the lower are directed downward and outward. Let's summarize our lesson for today. The layers of the retina include the retinal pigmented epithelium, layer of the rods and cones, and we mentioned that this layer contains only the outer segment of rods and cones. Then the external limiting membrane, outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglion cell layer, optic nerve fiber layer, and finally internal limiting membrane. Let's compare between the two types of the photoreceptors, rods and cones. The number of rods is about 120 million, while cones 6 million. The distribution in retina, the rods mostly peripheral in position, while the cones mostly central. Shape of the outer segment, the rod have rod shape, cylindrical, while the cone have conical shaped outer segment. The membranous discs not continuous with the cell membrane, while that of cones, the discs continuous with the cell membrane. The outer fiber long, while cones short. The synaptic region spherule in rods, while that of the cones appear like a pedicle. The visual pigment of the rod is the rhodopsin, while for the cones is iridoxine. The rod cells are responsible for dim light, while the cones receptors are responsible for the color vision. The structure of the eyelid, it is a movable fold of tissue formed of skin without subcutaneous fat. The lid margin show the eyelashes. Then the skeletal muscle surrounding the eye, orbicularis oculi. Then dense connective tissue containing meibomian glands, which is tarsal plate, and finally the conjunctiva. This structure from the anterior part to the posterior part. Remember that the refractive media of the eye are four transparent structures through which the light passes to reach the retina. They are the cornea, aqueous humor, lens, and the vitreous body. Time for quiz. Which of the following is a receptor for color? Rods, cones, bipolar cells, ganglion cells, horizontal cells. Exactly, it must be photoreceptor cell, rods or cones. And for color vision, cones are responsible for color vision, while rods responsible for vision in dim light. Okay, so the answer is cones receptors, responsible for color vision. The region where the optic nerve exits the eye is called optic chiasm, lamina cerebrosa, optic disc, aura serrata. Exactly, it is a blind spot optic disc. Thank you so much for your listening.